In this video, we're going to talk about review part 1, focusing on cell transport, organelles, and ATP. So this is the first 2 pages of your final exam review packet. So the first thing we're going to talk about is osmosis versus diffusion. So we learned about osmosis and diffusion at the beginning of our semester. Diffusion is when you have high to low transport of molecules. So for example, if we have certain molecules that are very close together, we say this is high concentration. Now, what would naturally happen is these molecules will bump into each other, kind of like bumper cars, and then they're going to spread out. So this kind of way of molecules naturally spreading out because they bump into each other, this is called diffusion. This happens naturally. It does not require energy. So high to low concentration, no energy. That's diffusion. However, osmosis is just a type of diffusion. Instead of thinking about how the solute is moving, we're thinking about how water is moving. So let's take a look at the definition right here. It says osmosis, diffusion of water through a selectively permeable membrane. So this selectively permeable membrane is also very important because otherwise the solute, such as salt or sugar, dissolved in the water, will also be moving around. In this case, they are still moving, but they're not passing the barrier. We're only considering for osmosis how water is moving. So water will naturally travel from an area of high water concentration to low water concentration, because in the end, this is still diffusion. So it's still high to low concentration, as in not the number of dots, how close together the dots are is what concentration means, and no energy is required over here. And when you have high water concentration, in comparison, you would have low solute concentration. So for example, if we're looking at this particular image, on side A, we have a lot of dots. The dots represent the solute. So on side A, we have a high solute concentration. So then in comparison, we would have a low water concentration, right? The more salt, the saltier the water is, in comparison, the less water you have compared to the other side in terms of concentration. Now, when we're thinking about concentration, it's always a comparison between one side versus another side. So on side B, in comparison, we have a low solute concentration and a high water concentration. Because osmosis is how water is moving, naturally, the side where you have more water will naturally equal out the side with less water. So water will move from side B to side A to equal out the concentration on both sides. If you think about 2 cups of water, if you have really salty water versus not very salty water, how can you make it even out? And if the only way that you can manipulate the situation is by adding water, you'll have to add more water to the side that is saltier. So you can think of side A as really salty water, right? But you can't remove the salt. So what can you do? You can add more water. So water will move from side B to side A. There's still water moving from side A to side B, but overall, the direction of movement is going from side B to side A. And now, eventually, we will even out the dots, which means the concentration of the salt water is the same. We will now have more volume of water overall on the A side versus B side, but the concentration, if you were to taste the water, the saltiness will be the same. So what's the moral of the story here? Water can move from an area of high water concentration to an area of low water concentration, which is the same thing as going from a side where there's low solute concentration to high solute concentration. And if you were to use a different name to represent low solute concentration, that means hypotonic. And high solute concentration means hypertonic. So you don't want to equivalent hypotonic or hypertonic as the cell is going to shrink or expand. Hypotonic is simply describing a certain side of having low solute concentration in comparison to the other side, which is hypertonic, high solute concentration. So how does that apply in a situation where there are cells? So for example, the first one, we have hypertonic solution. In this case, we start off with a cell we put a cell into a solution. The solution is what's on the outside of the cell. That's the environment. It's a hypertonic solution, which means it has a high solute concentration. Hyper means high. Hypertonic means directly high solute concentration. So what does that mean in terms of water? That's a low water concentration. Okay, so when you see an image or if you see a description of a question, you want to first think about how does that apply to the solute concentration versus the water concentration. Now, if we were to look at the cell itself in comparison, the cell has a low solute concentration, which also means high water concentration. If you were to translate this language to hypertonic, hypotonic, the solution outside of the cell is hypertonic, like we said, but you can also say that the cell in comparison is hypotonic. Okay, so what is the result? If water is moving through the selectively permeable membrane, which is the cell membrane in this case, and the solute, it could be sugar, it could be salt, whatever it is not passing. The purple dots are not passing through the cell membrane. So in order for you to reach equilibrium, the water will have to move from the cell out into the solution. And as you can see, the result after the result after osmosis is the cell will shrink as a result of water moving out of the cell. And now we are at equilibrium. So once again, you don't want to think about the word hypertonic and immediately think the cell is going to shrink. That is not what hypertonic means. All that the word hypertonic means is high solute concentration. If it says hypertonic solution, that means high solute concentration in the solution. If it says hypertonic cell, it means high solute concentration again, but now it's inside the cell versus out of the cell. Okay, so let's take a look at the second scenario. We have a hypotonic solution. Hypotonic solution means the solution is hypotonic, low solute concentration, which is high water concentration. And if you have high water concentration in an area, in order for us to reach equilibrium, the water will have to move out of that area. But in this case, moving out of the area means the water will have to move into the cell because there's less water concentration in the cell. So you're thinking about in terms of the blank space, the amount of the concentration of the blank space is low because you have a lot of the purple dots. The purple dots represent the solute, right? If you have a lot of dots together, you have less space. Kind of like if you have a lot of people crowded together in a room, relatively every person has less room. There is less room in the room, even if it's a huge room, right? So this is the case. Well, how do we equalize the amount of room for the people compared to outside of the room? Well, if the wall can move, you can make the wall bigger and give it more space. So if you put a cell into a hypotonic solution, the cell itself is actually hypertonic. But either way, water will move from an area where there's higher water concentration, which is in the solution, into the cell. So now, as a result of that, the cell will expand and potentially burst open and break. But now we reach equilibrium. The last one is easy. If we have isotonic, so hypo means low, but low solute. So hypotonic solution means low solute concentration in the solution. Isotonic means equal concentration. Isotonic solution means equal concentration of solute. Once again, solute. But you can also shift your perspective and think water. So now in this case, the concentration of solute in and out of the cell and the concentration of water in and out of the cell are the same. The water is actually moving in both directions into the cell and out of the cell, but it doesn't change the overall concentration of the solute or the water. So now the concentration of both will still be the same. There's no net change of the cell size. The cell remains the same size. And there's no net movement of the water. As in overall, the water is not moving, even though the water is moving. But because we're moving in both directions equally, there's not an actual change in the amount of water moving in one direction versus the other. So over here, you can think about if I were to ask you to draw hypertonic, hypotonic, isotonic solution, how would you draw it, right? So if you have hypertonic solution, you draw dots closer together out of the cell versus in the cell. And if I were to ask you to draw an arrow to show where the water is moving overall, more water is moving out than water moving in, and the cell is going to shrink. In comparison, you can draw a hypotonic solution. You put a cell in it, so you have a lot more concentrated dots inside the cell versus out of the cell. And the result of this situation is water will have to move into the cell. More water will move into the cell, and now the cell is going to expand for us to reach equilibrium. The last scenario is isotonic. If you were to draw this, you can just draw dots evenly spread out. And once again, we're not drawing the same number of dots, right? As you can see, I have clearly less number of dots inside the cell versus outside, but concentration really means how spread out the dots are. So in this case, water is moving in both directions, and the cell is going to remain the same size as it is before. And these are the answers to the review sheet questions, which you can pause and take a look and check your answers if needed. Okay, so we're going to try a practice problem over here. We are putting a cell into a solution. So the outside of the cell over here is the solution. Just to make it really clear, I'm going to write the word solution. And inside the circle, we have a cell. So in this case, it says we have 90% glucose. And you want to think about, is a glucose the solute or is it the solvent? The solute is what you dissolve into the water, and it can evenly spread out. So 90% glucose means the glucose is the sugar. You're putting a lot of sugar into this solution. It's very sugary water. And you put the cell into the very sugary water. What is that going to look like? And we're going to draw this to help us see very clearly. Okay, it's very important for you to draw this so then you can just have it right in front of your eyes and you can tell what to do with your question. So we have a lot more dots on the outside of the cell than inside of the cell, but more importantly, it's really not about the number of dots, it's about how spread out the dots are. Let's just make it really clear. Let me just put even more dots on the outside, closer together. So the idea here is that we have high concentration of the solute, okay? And in case you are confused, the dots are the solute and the empty space is the water. Okay, so now let's take a look at how we can answer this question. The percentage of water inside the solute, you just shift your perspective. If it's 10% glucose, and I'm not telling you, obviously a cell has all different kinds of other content, but we're just thinking, you know, a made up cell with only 10% glucose and everything else is water, 90% water. And then we have the percentage of water outside of the cell, which is 10%. And once again, osmosis means water moving from high to low concentration in terms of water. Because osmosis is the diffusion of water. And once again, diffusion is high to low. I see high to low, it's easy to drop things. High to low, no energy. It's the movement of water where there's more water to less water, concentration-wise. So you have higher concentration of water in the cell and lower concentration of water outside of the cell. So overall, if you're thinking about how water is going to move, water is going to move out of the cell and into the cell, but more going out of the cell. So if you were to just draw one arrow, just draw one arrow out of the cell. If you're drawing two arrows or multiple, draw the bigger arrows out of the cell than the ones into the cell. Okay, so osmosis will occur, how do I write this? From, let's see, water, oh, sorry. The question says, will osmosis occur? Why is it important to read the question? The answer is yes, water will move. And in what direction? Out of the cell. Okay, now in this case, the question didn't say, but we're only thinking about osmosis, so we're just assuming that glucose will not diffuse. And will the cell shrink or swell? If water is moving out of the cell will swell. The cell's getting smaller. How do you know? Because water's moving out of the cell. I'm just not gonna write it. The cell is in the what environment? So we're not thinking about the cell itself, we're thinking about the solution. This is the solution, okay? So the solution it has high concentration of solute. And it just happens that the word hypertonic means high concentration of solute. And once again, the word environment means we're talking about what's outside of the cell, which is the solution. The solution is high in concentration of solute, which correspond to the word hypertonic. Very good. The rest of the question says water moves from an area of hypertonic to, my bad, hypotonic to hypertonic, right? And this might be a little tricky. You really got to think about it. What do I mean by this? Hypo means low concentration of solute, which means high concentration of water. Hyper means high concentration of solute, which is the same thing as low concentration of water. So once again, water moves from, it's diffusion of water. So water will move from high water to low water, which is low solute, to high solute, which is the same thing as hypotonic to hypertonic. We're just saying the same thing with different languages. Why do we do that? I don't know. You shift your perspective. We're thinking about the scenario in different ways. J, the cell has higher concentration of, so we're thinking about the cell, what's inside the cell, which is right here, this circle, what's inside the circle, higher concentration of water and lower concentration of solute. Okay, your brain should be looking at the dots versus the empty space. The water is the empty space, right? There's a lot of empty space versus the dots are not very close together, so lower concentration of solute in comparison to the solution. So now the cell itself is hypotonic compared to the solution, okay? And once again, I'm saying this one more time, it's really important for you to know, hyper versus hypo is not about the cell shrinking or expanding. You really got to look at, am I talking about hypotonic cell, or am I saying the solution is hypotonic? Those are two very different things. All right, and here's the answer, keep the same thing. Moving on, we have a very simple question of looking at different parts of a cell. Obviously, there are more parts to the cell, but we're focusing on these parts for our final exam this year. So in this figure, it even gives you a word bank. How easy is that? A is on the very outside of the cell. We're looking at a eukaryotic cell first off. Tick. There we go. All right, we're looking at a eukaryotic cell because we can see the outside cell membrane, of course, because it's a cell. All cells have cell membrane. But inside the cell, we have a lot of these, what we call membrane-bound organelles. As you can see, they're like little compartments, right? They're like rooms inside of a room. We don't just have one big room as a factory. We have a school with rooms, and different rooms have different purposes and different setups. That's exactly how it is for the cell as well. You have these different compartments, different rooms to allow different reactions to happen and different processes. So we have A, which is the cell membrane. And obviously the cell membrane helps containing the cell and also allows certain material to go in and out of the cell. That's the purpose of the cell membrane, which you also should know. The cell membrane is a selectively permeable membrane, which means some material can go in and out, and there are different mechanisms to allow that. B is our nucleus. And the nucleus is where you store your genetic material, which is DNA for most organisms. The nuclear membrane, which is on the outside, is also a selectively membrane, the permeable membrane. All the membranes that we see, like all the walls that you see inside the cell, they're all, not the cell wall, but all these different membranes, they're all selectively permeable, which means some material can go in and out because we need to keep that room the way it's supposed to, right? It's like your room for Spanish class shouldn't have students who are learning German. That wouldn't make sense, right? We have the space set up for Spanish, and that's what happens. So that's what the membranes are for inside the cell as well. All right, we're moving on to C. C is meant to be the lysosome for the cell. So the lysosome is kind of the recycling bin for the cell. If there's some material that you no longer need in the cell, the lysosome will help you digest it, break it down, and then use it later at some point. The lysosome can also defend the cell and break down some of the invasions from outside of the cell. Maybe some bacteria that got taken in by the cell, for example. C is the mitochondrion, or if you make it plural, you can write the word mitochondria. Okay, so we only have enough space for mitochondria, so that's what I'm going to write. So the mitochondria or mitochondria are used to produce ATP. You don't just want to say mitochondria is the powerhouse of the cell, but what does that mean, right? The mitochondria are using the food that your cell receives, the nutrients, but really break down that nutrient and make ATP so that ATP can be used as energy for different cellular activities. For example, active transport. E is the smooth ER, smooth endoplasmic reticulum. You can tell it's the smooth ER and the rough ER, not the Golgi, by looking at the location and what it looks like. So in terms of location, the smooth ER right here, the purple one, is right outside the nucleus and is connected to the rough ER. But in terms of the purpose, the actual function of the two organelles, they're very different. The smooth ER is for detoxify, having a moment of not being able to spell, detoxifying the cell and also produce lipid. So what we mean by detoxifying the cell, if there's any kind of poisonous chemicals that got taken in by the cell, the smooth ER is in charge of breaking down the chemical and ensure that your cell stays alive. It also produces lipid for a variety of reasons, lipid hormone, other types of lipid, low battery. So those lipids are also produced using the smooth ER. The rough ER, however, is rough because it has these ribosomes on it. We can also find the ribosomes floating around inside the cell, as you can see these little dots. But the ribosomes on the rough ER, just like the ribosomes around the cell, helps you make proteins inside the cell. Okay, so they're different. The ribosomes that are floating around have also make proteins, but they're proteins for other purposes. You can just remember, for our purpose of the final exam this year, the ribosomes make proteins for the cell. That's good enough. But then some of those proteins, actually, they're very special. They might need to go out of the cell. Maybe the proteins will need to go into the lysosome. But before that can happen, the proteins have to be shipped over in a vesicle and then enter the Golgi apparatus. So the Golgi apparatus is in charge of packaging the protein. It can tag the protein, add certain chemicals to the protein, essentially telling the protein where it is supposed to go. Maybe it needs to go out of the cell. Maybe it needs to go to the cell membrane. Maybe it needs to go into the, stay in the lysosome and become a part of the lysosome. Either way, if a protein has certain destiny that's a little more complex than just your protein do the job, maybe it needs some further modification, and that is done in the Golgi. Okay, so these are certain organelles that you need to focus on for the final exam. And here's the answer key. Some additional information that you need to know. The first one is called the theory of endosymbiosis and the example. So the theory of endosymbiosis is talking about a big kind of prehistorical eukaryote. And then there's another little prokaryote. Okay. And what happened in the prehistoric time is that the prokaryote kind of got eaten by the eukaryote. But normally when a cell gets eaten, it gets digested, broken down, and that's the end of the little cell's life. However, in this case, the eukaryote was benefiting the prokaryote, and the prokaryote was coexisting and benefiting the eukaryote. That's kind of where the word the symbiosis is coming from, the symbiotic relationship where they're living closely with each other and they're, in this case, benefiting each other as well. This prokaryote was able to divide inside the eukaryote, and when another eukaryote is made as the next generation through asexual reproduction, we now also have another eukaryote now having these prokaryotes in it. But what are these prokaryotes? Why are they important? These prokaryotes eventually not only stayed inside the cell, they're no longer bacteria, and they evolved into the mitochondria and the chloroplast for cells. So mitochondria exist in all animal cells, fungi, plants. Chloroplast exists in fungi, certain fungi, and also plants. So the theory of endosymbiosis, in terms of examples, it will be mitochondria and chloroplast. We say it's a theory. There's certain evidence that tells us that this is something that we think might have happened that allowed mitochondria and chloroplast to exist nowadays. And that's what this theory is about. rough ER, smooth ER, all these functions we talked about already. Structures that plant cells have and animals don't. We want to focus on two, which are chloroplast and also, not mitochondria, the cell wall. The plant cells also have a large central vacuole. Animal cells have smaller little vacuoles, but these are two that we are focusing on right now. Okay, here's the answer key for certain questions. You can pause and take a look as needed. And the last two parts of the question is a comparison of prokaryotes versus eukaryotes, and also ATP versus ADP. So prokaryotes versus eukaryotes. Prokaryotes do not have a nucleus. This is, I'll just highlight it. This is also important because you want to think about prokaryotes don't have those internal membranes, right? Here's what a prokaryote looks like. You have the cell membrane, there's a cell wall. It might have a flagellum, it has a little circular DNA, and some ribosomes. There are other things in the prokaryotes, but that's what you need to know right now. A eukaryote, we saw the picture earlier, is very complex. So eukaryotes, on the other hand, do have a nucleus. So the DNA, instead of just floating around in the cytoplasm, the liquid environment, this is the cytoplasm, they're housed in a nucleus. Is it big or small? Smaller, bigger. Less complex, more complex. As you can see, the eukaryote has all those different compartments, some of which we didn't even talk about. Very complex, okay? There's a lot that needs to go on in the eukaryotic cell than there is prokaryotically. But a prokaryotic cell is also very, it's simpler, but it does the job that it needs to do, right? You might have a room that's just the bathroom versus the Spanish classroom, right? The Spanish classroom is more decked out and has a lot more activities going on, different types of activities, but it serves its own purpose that it's supposed to. So prokaryotes, eukaryotes, there's not one that's more evolved than the other. They're all advanced the same way as each other. They fit their own environment, works out. Does it have a cell membrane? If it's a cell, it has to have a cell membrane. Otherwise, it's just leaky water. Does it have a cell wall? Prokaryotes all have cell walls. Eukaryotes, you can say yes or no. Well, yes will be a better answer, but really the answer is to not write with a highlighter. Sometimes plant cells have a cell wall. Certain fungi have cell walls. Animal cells do not have a cell wall. Does it have DNA? Yes, they all have DNA to serve as its genetic information, genetic material. The shape of the genetic material is circular, but still a double-stranded DNA. But the double-stranded DNA twist is a circle, versus the eukaryotic cell has many linear chromosomes DNA. This is one circular DNA. Does it have a membrane-bound organelle? No. The membrane-bound organelle are the internal membranes that we were talking about earlier. The prokaryotic cells only has the external membrane that allows certain things to go in and out, all that. But inside the cell, it just has the cytoplasm, ribosome, DNA. Versus the eukaryotic cells, they all have those internal compartment, the nucleus, not ribosome. Ribosome doesn't have a membrane on the outside, it's just one thing on its own. It doesn't have an internal environment, but the Golgi, rough ER, smooth ER, lysosome, vesicles, chloroplast, mitochondria, they are all membrane-bound organisms. They're rooms inside the bigger building. What type of organisms have this type of cell? These are bacteria, and archaea. These are the prokaryotic cells. Eukaryotic cells, we have plants, animals, fungi, and protists. Protists, which I don't have room for. Are there membrane bound organelles? And that's a repeated question. This is still no, this is a yes. Organelles that are common for both, you really just want to think about what's in a prokaryotic cell. If a prokaryotic cell has a cell membrane, the eukaryotic cell has it. The prokaryotic cell has a DNA. It's not really an organelle, but it's still a cell compartment. So we're emphasizing it here that everybody has it. Eukaryotic cell also has DNA. Ribosomes are important because all cells need proteins. Proteins allow molecules to go in and out of the cell transport. Proteins are just in the cell membrane in general. Proteins allow, enzymes are made out of proteins, so enzymatic reaction, certain chemical reactions require those proteins. So you need the proteins in all the cells. And where do you get the proteins? The ribosomes are in charge of making the proteins. And lastly, it's not the hot Sahara desert. We need the liquid inside the cell. The cytoplasm is the liquid with all the proteins and other chemicals inside cytoplasm. Everybody has it, okay? Here's the answer key. We just saw it. Lastly, we have a comparison between ATP and ADP. The entire answer is already shown to you, but let me explain it real quick. On the bottom, we see ATP. I just want to emphasize. Let me really write it right here. The whole thing, this whole thing is ATP, okay? On the top, we have ADP. The ATP is called ATP because it's standing for, the T stands for triphosphate. Tri, as you know, means 3. And there are, in fact, 3 phosphate groups. Versus ADP, D stands for diphosphate. Di means 2. Okay, so what is the big deal over here, right? Why do we always talk about ATP as if it's the star child? And it is. ATP is the energy that we can use inside our cells. It's great that you eat your burgers, you're getting your protein intake, you want a little bit of fat in your body, or whatever. You have to need a good amount of lipid, fat inside your body to feel alive. You know, eat your food, eat a balanced diet. You need your carbohydrate, your sugar, those are all good for your body, right? But all of that is fantastic, except none of it can serve your body directly. Your cell cannot use sugar directly. It's too big of a molecule. So your cell will actually turn that sugar into ATP. So that's cell respiration. We'll talk about that. ATP holds energy because the 3 phosphate groups, they all have negative charges and they're very unstable. It's like putting 3 dogs that hate each other into the same room. They're going to be barking and fighting, and it's just not a good scene, right? There's a lot of tension. That tension really is chemical energy. It's being held right here in this bond right here between the 2nd and the 3rd phosphate group. So how do you release that tension? How do you release that energy? You will break this bond, which allows you to release the energy and also release the phosphate group. That's how you release the energy. So now you can think about ADP kind of as a dead battery, right? It's not dead. It still has potential energy, but now you can recharge it. Add on that phosphate group, add that extra phosphate group, and now you have ATP again. So this is an ADP-ATP cycle. You make ATP for your cell uses the ATP, and now you have ADP left over, and then you can recharge the ADP and make ATP again, okay? So that's a whole cycle. Don't just think up or down. I can put ATP on the top, ADP on the bottom, same answer, okay? So that's all for part 1. I hope this is helpful. If you have any questions, you know where to find me. That sounds a little creepy, but either way, good luck. I'll see you for part 2.